It's really a pleasure to be talking today about how we can optimize theta burst stimulation for clinical applications. So my name is Sara Tremblay. I am a scientist at the Institute of Mental Research in Ottawa, also assistant professor at the University of Ottawa. I had quite a bit of experience using theta burst stimulation and got trained at UCL by John Rothwell in its use. So here's the outline of today's presentation. So I'll first be presenting the technique. So what is theta burst stimulation and what is its main principles of application. And I will then follow on the clinical applications of theta burst stimulation. I'll be talking about specifically major depressive disorder because this is my main area of focus, but many of the things I'll be discussing about can be applied to other clinical populations. I will then introduce how two main techniques can be used to optimize theta burst stimulation. The first one is TMS-EEG, and the second one is neuroimaging. And finally, I'll be giving a bit of a summary of what was discussed today and talk about some future directions, some thoughts about the future research. So please don't hesitate to ask any question during the presentation. I'll be very happy to pause and answer them. So first of all, what is theta burst stimulation? So as a bit of background, I think it's important to understand the history of TMS before talking about how theta burst stimulation was introduced. So magnetic brain stimulation is based on the principle of electromagnetic induction that was first discovered by the English physicist Michael Faraday in 1831. However, the years that followed that discovery were mainly focused on electrical stimulation and not magnetic stimulation. It's about a decade after that Bickford and Freeman showed that it's possible to show muscle contraction following a magnetic stimulation of the nerves. And finally, it's in 1985, that Barker and colleagues did the first successful magnetic stimulation of the brain, where they stimulated the human motor cortex and showed that you could induce a muscle twitch in the contralateral arm. A few years later, there's been some improvement in the TMS devices, and the first devices that allowed for repeated stimulation, or rTMS, were introduced about in 1989, that we started to be able to stimulate at certain frequencies, brain areas. So now what is rTMS? So theta burst stimulation is a form of rTMS. So before talking about theta burst, we need to all understand what rTMS is. So rTMS is basically when we use TMS in a repeated fashion. And rTMS can induce after effects that outlast the period of stimulation. So that's what's very specific about it. And we think that those after effects potentially involve plastic changes in the excitability of cortical synapses. So the effect of rTMS can be measured in the motor cortex. I'll try to get the pointer so I can show. Okay. So the effect of rTMS, as I was saying, can be measured in the primary motor cortex, and where we measure the amplitude of the evoked muscle activity through the motor evoked potential. So typically, when we send rTMS at a high frequency, so over 5 Hertz, what we observe is an increase in cortical excitability. So here, an increase in motor evoked potential following stimulation. When we apply rTMS at a low frequency, so typically 1 Hertz or below, we observe a depression of cortical excitability. So these are the two main rTMS measures that are used. Now, theta burst stimulation was introduced 15 years later, so in 2004-2005 by Wang and colleagues. And it was really introduced in an attempt to mimic the technique that is used to induce plasticity in hippocampal slices, so in animal studies. The basic element of theta burst stimulation is a 3-pulse burst that we can see here that is given at 50 Hertz every 200 milliseconds, or 5 Hertz, which slightly differs from animal protocols where typically a 4-pulse burst is given 100 hertz every 200 milliseconds. So they had to adapt a little bit the paradigm given the capabilities of the TMS machines at the time. So theta burst stimulation does differ from rTMS in several ways. So with rTMS, we're talking about the frequency of a pulse that's given that's going to determine the after effect on brain excitability. With theta burst stimulation, we talk about the pattern of stimulation that's going to determine the activity, the after effect of the technique. Also, one particularity of theta burst stimulation is that it's given at sub-threshold intensities. So typically at 80% of the active motor threshold, which is quite low, whereas rTMS is typically delivered either at threshold or above threshold, so about 100% of the resting motor threshold to up to 120% of the resting motor threshold. So this is also one major difference between both paradigms. So as I said, the pattern of stimulation of theta burst stimulation will determine its after effect. So we can see here below the two main theta burst stimulation technique. The first one is the ITBS, or intermittent theta burst stimulation, in which we're giving a short train of 10 bursts that's going to last 2 seconds, followed by an 8-second break. This is repeated 20 times for a total of 600 pulses. And ITBS has been shown to increase excitability in the motor cortex. Now, continuous theta burst stimulation, or CTBS, is given continuously for 20 or 40 seconds, so for 300 or 600 pulses. And it is known to decrease the excitability in the motor cortex. So it's thought that the mechanism of action, the conceptual mechanism of action of theta burst stimulation, is that it produces a mixture of facilitatory and inhibitory activity, and that by giving ITBS in an intermittent fashion, we can keep the excitatory activity as predominant, whereas by giving theta burst stimulation in a continuous fashion, we're keeping the weaker inhibitory activity predominant, which determines the after effects. So although these bidirectional effects are well established, which is ITBS increases, CTBS decreases activity, there are still some important factors to consider when using theta burst stimulation. And these have been found from several studies in the motor cortex that looked into some technical aspect of the technique. The first one is inter-individual variability. So although on average, after ITBS, for example, we'll see an increase of excitability, it's not everyone that's going to show this increase. So about 40 to 50% of individuals will respond in the expected direction. And this is quite important to keep in mind when talking about clinical applications of the technique. The second important factor is there seems to be nonlinear effects of at least of duration. So what studies have shown in the motor cortex is if we prolong the duration of ITBS and CTBS, so if we double the duration, we actually see a reversal of the effects. So ITBS becomes inhibitory and CTBS becomes excitatory. Also, some studies have looked into the repetitive application of TBS, so applying several blocks of TBS. And it seems that the interval between blocks is crucial. So there's a specific interval for ITBS and a specific interval for CTBS that needs to be done to see the after effects that we're expecting. So for ITBS, it seems that the best interval is 2 minutes, whereas for CTBS, it appears that it's 20 minutes. So these factors, I think, are very crucial when we think about clinical applications. So now, as I said, so this was a very brief introduction of theta burst stimulation. I think I could have talked only about the technique for 1 hour, but I just wanted to give a quick overview of the technique to get into the topic of clinical applications. So as I said, I will focus on major depressive disorder, also because there's been the most studies with theta burst stimulation with that population, but a lot of what will be discussed can be applied to other clinical populations, and I'm happy to answer to questions about other clinical populations as well. So I do like to go back a little bit in time and see how techniques were brought up. So in parallel to its use in neurophysiology, TMS quickly gained the attention of psychiatrists and clinicians for its potential to act on brain activity for depression, at least in a similar fashion than ECT or electroconvulsive therapy that was that's still used for treatment of depression. So in the 90s, following some studies that showed an imbalance in prefrontal activity in depression, there's been a series of studies that looked into the effect of rTMS applied to prefrontal regions on depressive symptoms with quite positive results. Then in 1998, after researchers tried different parameters, different intensities, and after there was a few reports of seizures, the first safety guideline on the use of TMS and rTMS were published. In the following years, there was a big interest on the clinical application of rTMS in depression, which led in Canada in 2002 to the approval by Health Canada of the technique for treatment of depression. And in the States in 2008, the FDA approved rTMS for treatment of depression. It took about 5 years after theta burst was brought up in 2005 before we saw the first case studies of theta burst stimulation in depression. And the first clinical trials were published in 2014, which showed promising results for the use of theta burst stimulation for treating depression. And it's in 2018, following a large clinical trial that was published by Blumberger and colleagues in The Lancet, that led to the FDA approval of theta burst stimulation in the US for treating depression. So, as I said, first rTMS was used for treatment of depression, and the way theta burst stimulation has been used is based on how rTMS was used or is still employed for treating depression. So the typical protocols for rTMS is either applying 10 hertz to the left DLPFC, so high frequency left, low frequency right, so 1 hertz applied to the right DLPFC, or what we call bilateral sequential, so applying to the left and then to the right in a sequential manner. So in a very recent evidence-based guideline paper by Lefauchard and colleagues, it's been shown that there is a level A of evidence for high-frequency stimulation to the left DLPFC, which refers to a definite efficacy. And there's a level B of evidence for both low frequency right and bilateral, which corresponds to probable efficacy. So the typical duration of rTMS treatments are about 30 to 45 minutes daily, applied daily from Monday to Friday for 4 to 6 weeks. So it's quite time consuming, it's quite demanding for the client. And it appears that the remission and response rates are of about 30 to 35% for remission and 40 to 55% for response. And I would like to bring your attention to the 40 to 55%, which is very similar to what we observe in the motor cortex for following stimulation with theta burst stimulation. So it appears that also for clinical application, not everyone will respond to the treatment. So as I said, around in 2014, the first clinical trials were published. And we've decided to do a meta-analysis of sham control studies that we published in 2017, we wanted to see if there was an average efficacy of theta burst stimulation for treating depression as of then. And what we saw is that there seemed to be efficacy of theta burst stimulation when compared to sham, and that the most efficient at the time protocols were ITBS to the left and bilateral sequential, with TBS to the right, showing the lower efficacy. And here I just wanted to show that ITBS or theta burst stimulation is used in the same way as rTMS. So we apply the excitatory protocol to the left, the inhibitory protocol to the right, or sequentially in a bilateral fashion. So one thing, though, that we observed in 2017 was that there was not many studies that compared theta burst stimulation to rTMS. So we didn't know which one was more efficient or if they were equally efficient. But one major advantage of theta burst stimulation is its duration. So ITBS is only 3 minutes. CTBS is only 40 seconds. When we do bilateral, it's about 4 or 5 minutes. So it's much shorter than the daily sessions for our standard rTMS. So even if it was just equally effective, that would be very good use. The other advantage is that we stimulate at a lower intensity, like I mentioned at the beginning of the presentation. So with theta burst stimulation, we stimulate at about 80% of the active threshold, which is potentially makes it more tolerable than rTMS for clients, especially those with higher threshold. And then in 2018, that's exactly what some researchers did. So they directly compared rTMS and ITBS, so 10 Hertz and ITBS to the left, the LPFC, in what is the largest clinical trial that was published, so over 400 patients that were randomized either standard rTMS or ITBS. And this was a non-inferiority trial. So basically what they showed is that ITBS is non-inferior to rTMS, so it's equally effective. And we can really see from the two curves. So this is they deliver treatment for 6 weeks. So you see the decrease in symptoms that is equal between both treatment. So that was really good news. So we knew that rTMS, at least rTMS specifically, seems as effective as standard rTMS. This large study also allowed to tap into some important questions, such as what are the factors that influence response to rTMS or ITBS. And what they showed, which was quite interesting, there seems to be 4 trajectories. One of them is non-response, so people that do not respond from the beginning. And what they showed is those people that did not respond had a higher baseline score that we can see here, and also we're using benzodiazepines, so which seems to be shown that seems to be associated with non-response. There was also another trajectory, which was a fast response, which was associated with older age, a lower baseline score, and no benzodiazepine use. So it appears that benzodiazepine limits the efficacy of rTMS and ITBS, potentially by acting on the capacity of the brain to display plasticity. In parallel to this large trial, there's been some smaller published trials that looked into what we call accelerated TBS protocols, so or high-dose theta burst stimulation. So in those protocols, because theta burst is very short, people started to think, well, why don't we apply theta burst multiple times a day for a shorter period of time for the same amount of total amount of sessions? So studies have shown that it seems promising to reduce symptoms of depression. So studies have used from a number of sessions ranging from 3 to 10 per day. And so there needs to be larger trial with accelerated TBS protocols, but it might be effective for certain subpopulations of such as people with very severe depressive symptoms that need a very rapid decrease in symptoms. However, when I started working in the clinical application of theta burst stimulation, coming for a more neurophysiology side, working on the motor cortex, I was a bit shocked by the large variability in the number of pulses per session. So I did put a white background here just to warn you that the next slide is pretty busy. But what I want to show is basically all the studies that have been published so far that looked into the effect of ITBS and depression. And I want to bring your attention to the yellow numbers. So if I bring you back to the beginning of the presentation, I've discussed the fact that theta burst stimulation is applied at 600 pulses. So this is the standard stimulation. However, in clinical studies, the number of pulses have been increased significantly. So we can see that 1,800 pulses have been applied very frequently, and even 3,600 pulses per session. So to me, that was a bit concerning, especially given the fact that studies in the motor cortex show that increasing the number of stimulation pulses does not necessarily lead to better outcomes on brain activity. I observed the same thing in terms of the intensity of stimulation. So typically, theta burst stimulation is applied at 80% of the active motor threshold. However, we go back to the slide and now we look into the black numbers. We can see that, yes, there's been a few studies that use the standard 80% of active threshold, but also a lot of studies that use much higher intensity, such as 120% of resting threshold, which was used in the large trial. And it seems to be now the gold standard. So this was, again, a brief overview of the application of theta burst stimulation in depression. And I wanted our attention to focus on the current challenges. So one of them is that there's no way to actually test those parameters exactly where we stimulate. So to go and test those parameters in the DLPFC. So everything is tested in the motor cortex. And there's also no established predictors of outcome or marker of treatment efficacy. So most studies use symptoms as a marker of clinical efficacy, which makes total sense. But there's also, that would be also interesting to see, to track what are the effects of treatment on brain activity. One way to potentially track those changes is TMS-EEG. I just want to move my pointer so it's not in the way. So there's been a webinar on TMS-EEG, so I don't know if some of you have seen it by Nigel Rogash. But I'll briefly go over the technique. And I want to talk about two things. So the first one is how TMS-EEG can be used to select optimal parameters. And the second one is how TMS-EEG can be used to track changes in the brain in clinical trials. But now let's talk about selecting optimal parameters. As I mentioned, most parameters, if not all parameters, are tested in the motor cortex because of the motor output that we have that is quantifiable. But we don't fully know if what we test in the motor cortex applies to other regions, such as the DLPFC. So one example is the same intensity required, is the same number of pulses required to obtain changes in other regions of the brain. I actually started being interested in working in psychiatry when I saw the technique of TMS-EEG, because I wanted to have an objective output to be able to test those parameters in the prefrontal cortex. So TMS-EEG was developed, well, EEG, electroencephalography, is not a new technique. But before the last few years, when we would apply a TMS pulse on top of an EEG cap, the amplifier would just saturate, so it wouldn't be possible to measure, to record the activity of the brain. But with progress in amplifiers' capabilities, it's now possible to stimulate the brain on top of the EEG cap and to measure directly, to quantify the EEG response following a pulse. So it's really, to me, very similar to the motor evoked potential. So we can look at the effect of theta burst stimulation, for example, by looking at TP activity before and after stimulation, in the same way that we do it in the motor cortex. So when we send a pulse, what we observe is a TMS evoked potential, or TEP, which is which is a waveform that's characterized by a series of peaks and troughs. And really, these peaks and troughs have now been well characterized. So there's the P25 or P30, the N45, P60, N100, and P185 or P200. So these are the main peaks that we can observe following a single pulse of TMS. So TMS-EEG is a very interesting technique because it offers a neurophysiological index of the after effects of theta burst stimulation. So apart from the TMS evoked potential, or TEP, that I mentioned, we can also look into cortical oscillations following a TMS pulse, but also how EEG activity is spread over the brain, and looking into some measure of effective connectivity. Another interesting point is that it bypasses sensory and attentional processes. So the person can be just sitting, we stimulate the brain, you do not have to get involved in a task or go into a scanner. So it has a lot of applications. So for example, we can measure TMS-EEG in the brain before and after an intervention, exactly where we intervene. So if we do theta burst stimulation in the left DLPFC, we can measure activity in the left DSPFC as well, and then look into changes in the amplitude of our TMS evoked potentials. We can also look at TMS-EEG features as potential biomarkers that tap into the pathophysiologies of some clinical disorders. And we can also correlate some TMS-EEG features with behavior. So for example, correlating a change in TMS-EEG features with a change in clinical symptoms to try, again, develop markers of treatment response. So Chung and colleagues from the Fitzgerald group were the first ones to look into TMS-EEG as a measure of ITBS, of theta burst stimulation applied to the DLPFC. And although no effect was found for CTBS, they showed that ITBS modulates TEP amplitudes, so specifically the N100 and P200 here, and also modulate TMS evoked oscillations in the theta band. So that was the first demonstration that TMS-EEG could be used to study ITBS, or theta burst stimulation, in the DLPFC, which to me was very exciting. And they did a lot of very interesting follow-up studies where they looked into specific TBS parameters and studied them using TMS-EEG in healthy controls. And what they showed, for example, is that 2 blocks of ITBS, so of the standard 600 pulses applied at a 15-minute interval, actually does not produce stronger effects than only 1 block. So that has a potential application for those accelerated protocols in which the intervals between each block has been varying a lot between studies. So there needs to be some studies that look into the optimal interval, the optimal number of blocks that we can send to increase the effect of ITBS. They also look into different intensities. And so they looked into 50% of the RMT, 75% and 100% of the resting threshold, And just so we have a bit of an idea, 80% of the active threshold, which is used in standard theta burst stimulation, equals to about 70 to 75% of the resting threshold. So this was more or less the standard ITBS, and with a higher intensity or a lower intensity. And they did show a U-shaped relationship between intensity and after effect, with the 75% showing a stronger effect and 50%, 100% showing almost no effect. So one thing that I wish they would have done, but this was probably done in parallel to other studies, is looking into 120%, because this is now used clinically, and no study to date has looked into if maybe there's a U shape and then it comes back up at 120%. We don't know that yet, but TMS-EEG appears to be very sensitive to measure those effects. So as one of the first projects that I've conducted when I started working at the IMHR is looking into the optimal duration of theta burst stimulation for DLPFC stimulation, where we looked into the 3 different stimulation duration that were used clinically, so 600, 1200, or 1800 pulses. And we measured TMS-EEG before and after. So what we've shown, which to me was not surprising, is that longer is not necessarily better. So all 3 conditions yielded comparable effects on TB. So none of them was superior. 600, 1200, and 1800 showed the same effect. So what we showed is that, so in green, you have pre-theta burst stimulation, the average of all 3 conditions, so the average of the 3 duration, and in blue, post-theta burst stimulation. So we did show an effect on all components except the N100, which was quite strong, so reduction in the amplitude. We also looked at TMS oscillations, and same thing, we also showed that all 3 conditions yielded similar results. So there was no difference between conditions. And what we saw is that theta burst stimulation did reduce the power of theta oscillations and also modulate theta oscillation when paired pulse stimulation, so long-term, long-interval intercortical inhibition was applied. So again, suggesting that by using different index of TMS-EEG, we could really look into the effect of treatment duration. And it was quite clear that there's no advantage to increase stimulation duration. Now it would be interesting to do that same study with people with depression to see if the depressive brain reacts in the same way, but at least it suggests that 600 stimulation would be as efficient as longer stimulation protocols. So now, how can we use TMS-EEG in the study of the clinical effects of theta burst stimulation in depression? So to this date, there's been only one study that explored the effect of theta burst stimulation in a single case study using TMS-EEG. And what they show in that case study is before bilateral theta burst stimulation, so they applied ITBS to the left DLPFC, what they showed is that there was an imbalance in cortical oscillations before stimulation, which was reestablished after stimulation. So that was the first indication that TMS-EEG was sensitive to theta burst stimulation applied clinically. There's one study that looked into a bigger sample, but this time using rTMS in depression, that looked into 2 markers of TMS-EEG that are quite new, which is the current density, which is the first marker, and look also into connectivity. And what they showed is that at baseline, people with depression had a significant hyperactivity in the subgenual cingulate, which for a while now have been shown to be involved in depression. So it's one of those limbic structures that seems to be hyperactive in depression. which was shown by neuroimaging. But for the first time, we could show that this type of hyperactivity in deeper structure could be measured with TMS-EEG. And following treatment, they showed that this hyperactivity was reduced in people getting the real treatment, whereas it was not reduced in people getting sham stimulation. They also showed that connectivity between the left DLPFC and the subgenual cingulate cortex was increased following treatment. So it seems to be that the relationship between the DLPFC and the subgenual cingulate cortex is at least very important in the effects of rTMS in depression. So there's another study this time that didn't look at the effects of rTMS or theta burst stimulation, but looked at TMS-EEG as a measure of the pathophysiology of depression. So in this study led by Daphne Voyneskos, they show that people with depression have an increase in the amplitude of two main components of TEPs, which is the N45 and the P60. And even that the N45 could predict the mental illness. So it could predict people that had depression versus people that did not have depression. So it suggests that TMS-EEG has a potential also as a biomarker of depression, at least. So given all those studies, I was interested in developing a clinical trial that would use TMS-EEG as a main component. And I wanted to use TMS-EEG in three ways. So the first one was to use TMS-EEG as an objective marker of the effect of treatment. I want also to explore TMS-EEG as a potential biomarker of major depressive disorder to compare with healthy controls and see if we could have a marker in the brain. But also I wanted to see if we could predict individuals who responded to rTMS based on how the brain responded to theta burst stimulation at baseline. Because one of the main challenge, I think, is identifying who will respond to treatment early on, so they don't go into a 4 to 6 week treatment if they're unlikely to respond to treatment. So that, to me, is one of the main challenges that we are facing. So one of the novelty of that clinical trial is that when they first came in for their first theta burst session, we measure TMS-EEG before and after theta burst. So we have a plasticity index, and we're measuring that again at the end of treatment. So we want to see if there's a relationship between how plastic the brain is as baseline, but also if we're able to change plasticity as we go through the treatment. So this clinical trial is ongoing, although it's on pause right now because of the current COVID-19 situation. So patients are randomized either left ITBS or bilateral sequential because it was never studied in a large trial. And we're using the standard ITBS procedure, 80% of active threshold, 600 pulses. Hopefully, we can show in a large sample that this is effective, so we don't have to increase stimulation duration and intensity. The treatment is delivered for 4 to 6 weeks, and for people who respond, they move on into a maintenance phase. We also have a very specific maintenance protocol because I realized that maintenance was very few studies were looking into maintenance in rTMS and how it can help maintain the treatment efficacy. So we are also in the clinical trial studying 2 different types of maintenance. So now that I discussed a little bit about TMS-EEG, now I want to give an overview of how neuroimaging is helping us understand the mechanism of action of theta burst stimulation. So I see the time running, so I will briefly discuss 3 different types of neuroimaging techniques that I think are very interesting for studying the effect of theta burst stimulation. So the first one is magnetic resonance spectroscopy, or MRS. So MRS allows to quantify metabolite concentration in a specific voxel, so in a specific region of the brain. So there was a first study that looked into the effect of ITBS in healthy controls using using MRS. Sorry. So what they did is they had a voxel in the anterior cingulate cortex, or ACC, and the DLPFC, where they did stimulate with ITBS. And they specifically looked into glutamate and GABA, which are marker of excitation and inhibition in the brain. Because it's thought that GABA and glutamate are very important in plasticity mechanism and in the mechanism of rTMS and theta burst stimulation. And what they showed is after ITBS compared with placebo ITBS, there was a reduction in the balance between GABA and glutamate in both the DLPFC and the anterior cingulate cortex. So suggesting that these are very important in the mechanism of action of theta burst stimulation. There's been very few studies using MRS with ITBS in depression. I've seen one recent study that this time they looked into the anterior cingulate cortex only, and they measure metabolites before and after stimulation. Unfortunately, the sequence they used could not allow GABA to be measured. So they looked into other metabolites that are quite interesting, such as choline and NAA, or N-acetyl aspartate, which are thought to be marker of neuronal integrity, especially white matter integrity. And what they showed is there was a relationship between improvement of symptoms after a course of ITBS treatment in depression and an increase of the ratio of those two metabolites, suggested that potentially ITBS could normalize neural circuit function in the brain, especially in this case in the anterior cingulate cortex. But again, it showed that ITBS does not affect only the DLPFC. It does impact interconnected region, especially frontal limbic networks. And this relationship between DLPFC and frontal limbic networks have been shown with a resting state functional connectivity MRI. So in a series of studies by the Michael Fox group, it was shown that there is some hyperactivity in the anterior cingulate cortex, and there seems to be altered connectivity between the DLPFC and the anterior cingulate cortex in the brain of people with depression. And in the large study, they showed that connectivity between the DLPFC and the subgenual cingulate cortex could predict response to rTMS this time. So it suggested that potentially it could predict also response to ITBS because it has similar effect on depression. This was tested in healthy controls, and they showed that indeed ITBS can modulate functional connectivity between the DLPFC and the anterior cingulate cortex. This study was published just recently in 2020. In depressive patient, it was shown that that the response to accelerated ITBS protocols was also predicted by the connectivity with the subgenual cingulate cortex. So again, suggesting that there seems to be really a strong relationship between responders, so responding to a treatment, and the activity in that particular region. The last technique I want to talk about today is PET, or positron emission tomography. So when I first arrived in Ottawa, when I started working there, I was quite amazed by the new big tool that we had, which was a PET MRI, so the first dedicated PET MRI for mental health research in Canada. And I wanted to find a way to use that new toy, I would say. So I started looking into PET, and there's been and getting into contact with some PET MRI experts. And we did look into a review and meta-analysis of all published studies that try to understand the mechanism of rTMS using PET. What we've seen is a lot of variability. So we looked into studies that use rTMS on the left DLPFC, and here you could see all the different regions that are activated after stimulation. But when we did the meta-analysis, we did not see any consistent pattern of activity. Of note, in the review and meta-analysis, I have seen that no study to date looked into theta burst stimulation applied to the DLPFC in healthy control, so this still needed to be done. There's one study that used PET, measuring glucose metabolism after TBS treatment in depression, that showed, again, a very important contribution to the anterior cingulate cortex following treatment, so a reduction in the activity of the ACC, but also locally in the prefrontal cortex following stimulation. So because of the lack of healthy control studies, we started in collaboration with Larry Twiminen and Yuo Jutsa from Turku University. We looked into the effect of ITBS on the left DLPFC using glucose PET, so FDG PET, and resting state connectivity. So these are preliminary findings. So we were halfway through data collection, but now data collection is on pause. But what we've seen is with glucose, ITBS did reduce the concentration of glucose in the insula, the subgeneral anterior cingulate cortex, and the caudate. Similarly, looking at resting state connectivity, we saw a reduction in connectivity between the DLPFC and the insula and caudate. So again, it seems that for ITBS as well as rTMS, there is a key involvement of those limbic networks in the after effect of stimulation. So now, as a summary and future direction. So I think we can now confidently say that ITBS is an effective treatment for major depression, major depressive disorders. There are some current challenges that I have highlighted, such as trying to develop new tools to select optimal parameters, also developing ways to identify potential responders, and developing biomarker for prognosis. I hope I've shown today that I believe TMS-EEG has a future in clinical rTMS and theta burst stimulation in the field of clinical TMS as a potential marker of pathophysiology, but also to help with parameter selection and identification of potential responders. Neuroimaging, although expensive, it's also very useful, I think, to provide insight into the mechanism of action. So with neuroimaging, it's been shown that there's a key implication of limbic structures, such as the ACC insula and the neural effect of ITBS. It could be also a potential marker of treatment and a way to identify novel targets in the brain. So one of the future directions that I believe is very interesting is the emergence of novel targets. So you saw today I spoke only about the left DLPFC because this is the region that's been stimulated the most in clinical studies. But there are other regions that have been studied, such as the ventrolateral prefrontal cortex, the frontopolar cortex, or the dorsal medial prefrontal cortex. Some of the issues with those specific targets are that they can be more painful because they're closer to the eyes or closer to centrally. But again, maybe TMS-EEG could help us define best parameters to develop, to measure and to stimulate those regions. Another interesting aspect of TBS is that because it's short, it has the potential for multi-site stimulations. So one thing that I believe has not been explored in depression is stimulating more than one site. So because it's so short, instead of applying multiple blocks, why not stimulate more than one region? So I think this is something that future studies could explore. Another future direction is the use of novel technologies. So one of those novel technologies is the controllable pulse TMS, or the cTMS, that was developed by Road Research. So one of the particularities of the cTMS is that it can apply repeated stimulation using a monophasic pulse. So because of the limitation of TMS devices, single pulse TMS is typically applied with a monophasic pulse, but repetitive TMS is applied with a biphasic pulse, only because the TMS machine cannot send monophasic pulses as a high frequency. But the new CTMS allows it. And there's one study that was published in 2016 that showed this time it was low frequency, so 1 Hz TMS. And gray, this is what is a standard biphasic pulse. And actually in that study on the motor cortex, it didn't show any significant effect. But they showed that using a monophasic pulse could actually increase the efficacy of the stimulation protocol. So during my postdoc, I did a study using monophasic theta burst stimulation. And I was a bit optimistic. I not only wanted to use monophasic theta burst stimulation, but I also wanted to study another new idea, which was to apply theta burst stimulation simultaneously with other neuromodulation techniques, such as, in this case, transcranial direct current stimulation, or TDCS. So what we did is we applied TDCS during theta burst stimulation in 2 different directions. So one was following the TMS pulses in a posterior-anterior direction, and the other one was reversing the current. So the TDCS currents will go in the opposite direction of the TMS current. And what we showed is that when we use the current in the same direction, we could actually improve the effect of theta burst stimulation on the motor cortex. And reversely, when we apply the current in the opposite direction, we actually abolish the effect. So I recently received funding to now apply this new paradigm to the prefrontal cortex. So I hope that using theta burst stimulation with PDCS could potentially increase the after effect of treatment. And I'm going to be measuring this with TMS-EEG. So those are all new future directions that I think could be very interesting with theta burst stimulation. But I hope today I was able to give you a good overview of what's the evidence in terms of the clinical application of theta burst stimulation as of now. What are the tools that we can use to measure the effect of theta burst stimulation? And most importantly, how we can use these tools to actually improve our techniques and not be satisfied with what we have, but find new ways to deliver better treatments using neuromodulation techniques. So before, I just want to thank all the collaborators in my amazing lab, and I'm very happy to answer any questions.
Great, thank you, Sara, and sorry for interrupting you there. It's very
important
to ascertain when people will finish a sentence or not. So yeah, we've got plenty of questions coming in. You've very kindly allocated 20 minutes for questions, right?
Yes.
So there's quite a few that came in that I saved for the end. The first one is, does the laterality of rTMS and TBS vary with handedness? Or is it given to the corresponding DLPFC regardless of handedness?
That's a very good question. It does not appear to be related to handedness. So studies right now, they apply regardless of handedness, left FDSD stimulation as the main treatment. To my knowledge, it's never actually been studied. So looking into handedness and left or right stimulation. I guess one of the hypotheses is that handedness is very important for motor cortex stimulation, but probably less important when we talk about DLPFC stimulation. However, we don't know. So that's a very good future study here.
Thank you. So now there is another one regarding the response. Blumberger et al. 2018 showed that the symptoms were stable after the first week of the end of treatment, and Castor et al. Followed the patients for 6 weeks. However, on longer stretches of time, on the order of months, how many symptom patients regressed to their initial severeness of symptoms?
That's a very good question. Unfortunately, not many studies do follow symptoms, track symptoms in time following treatment. The few studies that did show that typically the symptoms are quite stable for about up to a year. And one thing that struck me when I started, when I was thinking about designing a clinical trial, is the big difference between what's done clinically so in private clinics or larger clinics, and what's published. So most private clinics do a maintenance protocol, but it's rarely published. So it seems that having some maintenance done can actually help having more stable symptoms over time. So this is why in the clinical trial that we're currently doing, we have a 6 months maintenance phase. And hopefully we can see with that maintenance how symptoms evolve and if they're very stable or if we can actually even improve further symptoms with maintenance.
Thank you. So there's another question now, and it's regarding safety limits and pulses. So hang on, I just lost the question. Safety limits for the total number of TMS pulses to be administered are suggested to be about 1,500 pulses. However, studies such as the Pratt et al. 2014 seem to apply 5 sessions a day with 1,600 pulses per session. Is it safe to apply such high doses?
Actually, there's a recent study that applied 18,000 pulses a day. So that's a very good question. And from those studies, it appears safe in a treatment-resistant depression population. But definitely, I think the next safety paper that's going to be published by TMS has got to look into those accelerated protocols. Definitely, I would not do that in healthy controls. I would be quite scared in terms of safety. I can see a little bit more why it would be used in a very people with a very severe depression as an alternative to ECT, for example. But we need to have studies with larger samples that really look specifically into safety, because I don't think we can just increase the number of pulses per day indefinitely like we're doing right now. So I think we need to take a step back and look into really carefully safety for those paradigms.
Great, thank you. I'm sure that answered the person's question. The next question, it's not necessarily a theta burst or rTMS question, but it's definitely important when you're administering the pulses, and that's the use of a AMT or RMT to calibrate the procedure. As somebody who hasn't started using TBS yet, I'm surprised at how different the approach to application is regarding AMT and RMT. Why do some researchers use RMT and others AMT, and without necessarily saying why in their methods section?
I totally agree. This is a super important point. I think, and I don't fully agree with that rationale, but a lot of people that do talk about rationale in their paper, they mention that in motor cortex studies, it was shown that doing the active threshold right before theta burst stimulation could actually have an impact on the after effect. But it makes sense to me because we're stimulating a motor region and we're activating the motor region right before. But with DLPFC, there's not much evidence. Well, there's no study that actually tested that. So I don't believe the rationale really stands. So I agree that I think one of the most important part is that we have like that clinical studies have some kind of an agreement on what's the gold standard in stimulation parameters. And this is why I decided, even though the large trial used 120% of resting threshold, I decided to take a step back and use the standard protocol, because I think it's important that we also have some large clinical trial that looked into 80% of AMT. And one of the reasons also, as I think that clinicians are used to their RMT, so this is also kind of a used to measuring the RMT before doing the stimulation. And also many clinical studies don't have EMG, so they do visual determination of RMT. And I don't think you can do that with, well, I'm pretty sure you can't do that with the AMT. It's impossible to look visually at the active threshold. So there is also a need to be a bit more rigorous and use, I believe, that we should use EMG to determine the intensity of stimulation.
Great, thank you. There's another question now regarding the clinical trial that you mentioned. What is the stimulation being used in the plasticity assessment session?
Sorry, I didn't follow this one.
Sorry, it was a very interesting clinical trial. What is the stimulation being used in the plasticity assessment session?
Okay, in the clinical trial. Okay, sorry, I was not. I didn't understand that point. So what we do in the plasticity assessment session is basically the same treatment. So we send TMS-EEG pulses to measure brain activity, and then we apply ITBS or bilateral theta burst stimulation, depending on what they've been assigned to, and we measure again with TMS-EEG. So I call it plasticity assessment because we look actually at the effect of theta burst stimulation right after the stimulation, and we don't wait, because almost all studies, well, all studies, they actually wait until the end of the treatment to measure the effect. I'm interested to see actually on day one, what are the effects.
Brilliant, thank you. Now there's a question now about the MR spectroscopy that you mentioned towards the end. When using multivoxel MRS, it is complicated to measure various metabolites. What has been seen to be a better marker, GABA or glutamate?
That's a very good question. It's a bit hard to answer because there's not many studies that have been done with theta burst stimulation or rTMS or depression. I know that a lot of studies have shown GABA alteration and GABA modulation, GABA alteration in depression and GABA modulation after theta burst stimulation, for example, the STAG group in the motor cortex. So if I had to choose, I would probably choose GABA, but I think glutamate is also very interesting. So I think from my understanding, most sequences can measure both GABA and glutamate at the same time, but it depends on the number of voxel. But yeah, if I would have to focus, I think both would be interesting, but I might have a bit of a preference for GABA.
Great, thank you so much. Really got 1, 2, 3, 4 more to go, if that's okay.
Of course.
And then I think we'll wrap it all up after that, because I think the 20 minutes will be up. Regarding depression studies, do you think that theta burst will be more useful for patients with depression who have cognitive deficits as part of depression, given that theta burst modulates hippocampi?
That's a very important question. I think there's, at least to my knowledge, not many studies that look at the effect of theta burst stimulation on cognition in depression. I think it would be very interesting to see if theta burst stimulation is more efficient in modulating cognition than rTMS, but it's never been done. In healthy controls, the data is a bit, it's unclear whether theta burst stimulation can modulate cognition. Some studies show more working memory improvement, but it's not super convincing. So definitely something to look into and a very interesting hypothesis that should be tested.
Great, brilliant. I actually miscounted because I don't know why. I don't know what's wrong with me today. I've only got 2 questions left.
Okay.
So this one now is about the number of pulses that are selected for a theta burst protocol. So why was 3 pulses at 50 hertz chosen? Why not 41 hertz or at 62 hertz, and why not 5 pulses?
That's a good question. I believe, well, the reason why it was chosen, I think they did as much as they could with the machine they had at the time. So they couldn't do 100 hertz. I don't know exactly. I would have to ask John Rothwell what exactly, why they chose specifically 50 hertz and 3 pulses. But I believe it's because of the limitation of the machine. So they tried to get as high as possible and as many bursts as possible, as close as possible to the animal paradigm that used 4 pulses at 100 hertz. So they were able to deliver 3 at 50 hertz.
Cool. That is what I think too. Now, last question. Do you think it's about closed loop TMS-EEG now? Just to give you the context we're going to it. Do you think when working with closed loop TMS-EEG, we can get a fine tuning for each patient? Then instead of using theta burst at 50 Hertz, we can use 54 Hertz, for example, and change the number of pulses in a train in order to have a more efficient and personal therapy?
Definitely. I didn't talk about closed loop stimulation today because there was so much to talk about, but it is a very interesting approach. I think I think it's super interesting, and definitely we should study more closed loop TMS-EEG and trying to individualize treatment. That may be one very important way to optimize stimulation. The only downside is we need to find ways to automatize a little bit more the process. So in the clinics, it would be difficult at the moment to be able to use to have that equipment ready to do closed loop TMS-EEG. But as the field grows, I think if we can develop something that's a bit more automated, it would be really interesting to use it in clinics.
Brilliant. So thanks for going through all the questions with us. That's all for them now. We really
appreciate
all the time that you've gone into giving the presentation and giving the people time to also have their questions answered. Loads of people are saying thank you in the Q&A as well. And I'd just like to thank you again for helping us out and doing this webinar with us. We really appreciate it. And we all think that this is a really vital talk that needs to be given how popular Theta Burst is. So thank you so much, Sara. We really appreciate it.
Well, thank you very much for the invitation. I'm so glad I could participate to those webinars. I know that people from MALab have been attending pretty much all of them. So it's really a great series of webinars. So thank you for making me be part of it.